Phew. You had a bad echo.
Yeah.
Is it better now? Also just move this. Okay. Is that better now? Cool. So as most of you know, there is two ways of categorizing TOS effects. So those effects that won't last more than the sonication period, which we will refer to classically as online TOS. So in this case, the neuromodulation is a few milliseconds, a few hundreds of milliseconds, and your readout is concurrent to the sonication. For example, if you're interested in MEP, what Tulika was presenting this morning. Other effects last longer, and I'm going to refer to those effects as offline TOS. And in this case, we usually apply more energy in the tens of seconds to a few minutes, and then we can record or have readouts that are that last for a few hours after the intervention. So in this case, you have plenty of time to put, for example, somebody in an MRI scanner for animals and observe ball-related task changes or resting state fMRI. And this is what I'm going to be focusing on today. Right, so if we believe that it is possible to induce neuroplasticity, then we can start drawing some hypotheses on the readout. And I'm going to be focusing here on MRI readout, okay? So imagine this is the area that you are going to sonicate. Here. And this would be the network associated with this area at rest, for example. So one thing that you might hypothesize is that perhaps you're going to see a change locally where you are neuromodulating. So for example, a change in bold. Another thing that you might expect to see is a different relationship between the sonicated region and the rest of the brain. So if the person is at rest, in change in the resting state connectivity. If the area that you are neuromodulating is hypothesized to have a causal role into a certain cognitive process, then perhaps you are also going to observe some task-related behavioral change. Related to those task behavioral change might be some task-related neural activity that you might also perturb. And in this case, the network would be the functional network, which would be different than the resting state network. And another thing that you might be hoping to observe would be, for example, chemical change in the neuromodulated region, for example, GABA change with spectroscopy. So most of the study that I will be presenting try to address those questions. Right, so the first study was performed in Oxford in collaboration with Jean-François Aubry, Jérôme Salé, Leonard Verhagen, Miriam here, the Oxford crowd, Matthew Rushworth, and that was in a non-human primate model. And in this study, we applied a 40-second train of pulses to either the perigenual ACC, the anterior cingulate cortex, or on different days bilaterally to the amygdala. Animals were anesthetized, and then we put them in an MRI scanner and recorded resting state fMRI. And what we observed, so we were looking at the co-variation between our targeted region and the rest of the brain. So this is a heat map, and it shows the areas that are strongly coupled with the ACC and those that are not so coupled. So if you are looking at this connectivity fingerprint of the ACC in baseline session and after intervention, you can see that you are strongly changing this network profile. And this is maybe more apparent here on the spider web looking plot, where you can see the strength of coupling between the ACC and a bunch of different other regions. And this was area specific. So if we were targeting another region, say bilateral amygdala, then we couldn't find any difference with the baseline session in terms of the functional connectivity, the ACC with the rest of the brain, which point at the specificity of the technique. it was exactly the same for the amygdala. I'm just going to go very quickly over it. So when TUS was applied, in the hours that were following, we could see a change in the way the amygdala co-vary with other brain region network. And just to flash out that actually, if you're looking at the extent of where in the brain was that difference maximum, it would be exactly where we were sonicating. And here I'm just showing the ACC results. Okay, so with this study, we have already evidence that it is possible with MRI readouts to see change that are quite long-lasting. We moved on to another study where we're looking at behavioral change. So this is a learning task where animals have to learn contingencies between object and reward. And once they've learned those contingencies, we produce reversals. And in humans, but also in the non-human primates, classically, the ACC will co-vary with counterfactual values and will allow you to switch to better alternative. So that's what we had in mind. So we first replicated what we saw in the previous study and even found on singular sessions, single days, that we would find these differences in connectivity profile of the ACC after TUS intervention. But I'm going to move now to the behavior. So that was in 18 different sessions. So yeah. And this is the reward probability over time of the different options. So as you can see, there are some options that are just better to select at the beginning of the task. And that's what monkeys do, and they're very good at it. They're very good at learning reward contingencies. They're very good at matching their behavior to the reward probability of different options. And already, if you look at the raw data after sonication of the ACC, you could see that things were a bit perturbed. In fact, they were still good at doing the task. Their global reward rate was still quite good. But it was, if you started looking at some idiosyncrasy in their behavior, that you could see the maximum difference. And what we were interested in was switching behavior. So we looked at their ability to switch to a better or to avoid a worse alternative on a future trial when they are in conditions when they don't have those better alternatives on the screen. And they're very good at it in the baseline session. But this was perturbed after the ACC was neuromodulated. So not only they were not that good at switching to the better alternative, but they were also more likely to choose the worst. And this was significant. Another way to look at exploration and exploitation in the animal research traditionally is to look at the relationship between entropy and cumulative state. So the more entropy you have, so the less information, the more you should be exploring. And that's exactly what we found in the baseline session. But this was abolished. You can't really clearly see it. My line is here. After the ACC was neuromodulated. So actually the ability, to extract information from cues and to exploit it in their behavior was perturbed. And this is the difference in slopes between the two regression lines. So it's quite a strong effect. Okay, so with this study, we do observe long-lasting, in the order of hours after the intervention, task-related change. In the third study, still in a non-human primate model, we looked at two prefrontal regions that are actually very close together in a little macaque brain. The area 4712O, which is right here, LOFC, and this is the frontal pole. And we know that the LOFC is linked with reward history, with this ability to parametrically co-vary with reward history and using it to choose options. And this is what I'm presenting here in this matrix. This represents to what extent previously rewarded choices are currently influencing your current choice. So you can see that you're very more likely to repeat a choice that has been rewarded in the past at trial T-1, but also T-2 and T-3. So it's decreasing, but it's still influencing your choice. So you're capable of maintaining reward history and reward information. And this was not true after the area 4712 was syndicated. So they were only capable of kind of maintaining reward information, but that happened very recently. So they were becoming more myopic in their behavior. This was not any different than the sham session after the frontal pole was neuromodulated. So we looked at the bold related, the associated bold response to this parametric representation of rewarded choice. And this is what we found. And that was also aligned with a lot of literature that, yes, if you're looking at Win-Status shift behavior or reward history and how that guides your current decision, you will find the LOFC activated. So that's what we found. We found an increased bold in those regions. If we looked at the difference between the baseline session and our control region, so if you have a map that is equally active, then you should see no difference, and that's what we observed. So this was equally active. And we saw a reduction particularly in those regions after applying TOS to this lateral OFC. And maybe it's more clear in those plot here representing a decrease in bold after those areas were sonicated. So here we have yet another evidence for another readout, which suggests that, yeah, neuroplasticity might be possible. Okay, and now, before I move on to human applications, I'm just going to very quickly, because we've mentioned this a lot this morning, highlight the fact that TOS is challenging in humans, particularly because we have a skull, and we have to quantify the skull somehow if we want to have efficient design, as Jean-François was saying this morning, but also if we want to be safe, because this is where the thermal rise will be surely maximum. And this is just a little preliminary results on some retrospective simulation that were done on published papers that show that actually, yeah, the maximum temperature rise will be in the skull. So it might be a good idea to quantify this. So this, and the ideal image to quantify, like Maria was saying and others this morning, quantify the bone is to get CT image, but they are not perfect because it involves exposing your healthy participants to ionizing radiation, so you may want to avoid it. So what we did in the lab, and this was led by my postdoc, C.T. Jakub, was to develop a deep learning method to go from one type of image to another type of image. And the idea was to go from a T1 weighted image because that's what you will have in the lab for neuronavigation anyway. So we were starting with the most common image that you would acquire for your NEEBS study and then get Huntsfield units from those. So she developed this unit. So it's a 3D convoluted neural network where you learn more and more complex feature of the image as you go down in the net. And then you're just putting this together to the space that you want, which is here a CT space. And the difference between this and maybe other convoluted network is that she used subpixel upsampling instead of the classic upconvolution, which is a little bit better for the checkerboard artifact. Okay, so to do this, we had this database where for each participant we had a low-dose CT and a 3D T1 weighted image. And to perform the deep learning, how well our deep learning was performing, we used the mean absolute error and the dissimilarity coefficient. And this is just a screenshot to show that, yeah, it is producing very nice looking pseudo CT, that our mean absolute error is quite similar to other methods that have been published. And the dissimilarity coefficient is about 82%, which is very good overall. And because we were interested in acoustic output, we also just compared our acoustic output on different measure from the real CT, the reference CT, our pseudo CT, and just a simple binarized mask, because that could also be an option that you could do to get bone information. And basically, we didn't find any difference between our acoustic output between the pseudo CT and the reference CT. And the binary skull actually did a pretty good job at in terms of trajectory. So there wasn't any, difference. That was maybe a good take-home message that actually even a binary scale is still good at telling you where perhaps the being will be. But it did not do such a good job in estimating the intensity compared to what you would get with a real CT scan. And this is also an open source project, and it does generalize quite well as long as the T1 image is relatively close to what was used to develop the deep learning. So yeah, please let us know if you're interested in using it. And this is my last study. I hope I'm not running too much over time. It should be okay. And yeah, I'm going to just very quickly talk about it because CT is going to present it more in detail tomorrow at 11. And Tristan also has a poster, so please go and ask them. So they both led this work, Rick. And the question here was, can we see a change in vivo concentration in GABA where we're stimulating? So we used a repeated theta burst protocol of 80 seconds before participants went into the scanner. So they were MR safe before the intervention. So we could very, very quickly put them in a scanner after intervention. We had a first run of resting state fMRI, 2 different voxels of MRS voxels and another resting state data there. People came 4 times. I have to highlight this because I'm very impressed with this. It was a repeated design, 24 participants came 4 times. And they came at the same time of the day and the same time of the week because GABA level might fluctuate. So the first time they came for a T1 weighted image that was used for neuronavigation and for the acoustic simulation. And then they came and they either had no intervention, so we still pretended that we were stimulating in the medial cingulate cortex, and or they had TOS intervention in the ACC, which is important for switching behavior, as you know, and the PCC because it's a very active node at rest. And this was fully counterbalanced across participants. This is just a result of how, well, the MRS voxel and how the acoustic field look like on overlay on top of it. I don't know if you can see it, but maybe this image is a bit more clear. So this is the focal volume, which is greater than the full width half maximum or -6 dB. And you can actually see that even though we were trying to optimize the voxel size as much as we can, but still having a good SNR given the we wanted to keep things short. So the voxel is 2 by 2 by 2, but still the focus is quite small actually within the MRS voxels. And there are 3 colors because these were 3 MRS for the 3 different days. And so in terms of data quality, we're pretty good, pretty happy with the error fit and the SNR. If anything, actually, the ACC was maybe had higher quality data than the PCC. And these are the results. Let me unpack this a little bit. So if you're focusing here on this right-hand side figure, this is the PCC voxel, this is the GABA level, and after sonication, there is a consistent decrease across participants. I mean, we were already seeing this at N15. This is very consistent. So there is a consistent decrease, which is not the case if the active region is stimulated. We didn't find any difference in the ACC. And similar to, I'm going to show some functional coupling difference in the PCC with the PCC voxel, but we didn't find any significant difference for the ACC either in terms of how the ACC co-vary with the rest of the brain. So, but we did find some very interesting, yeah, it's a little bit dynamic. So I'll let CT explain all of this tomorrow, but basically things seems to change over time. So after 15 minutes or after 50 minutes, there was different network involved. But overall, we saw an increase in coupling between the PCC that was indicated and many other regions. Yeah, so please come to us Tristan's poster if you want to talk about it, and City will explain this more in depth tomorrow. Why do we not see anything in the ACC? I think I'm obsessed about this question. But so one thing that we've learned, according to those data, is that actually the focal volume matter for MRS. So I mean, maybe because of the voxel size. So it looks like because the PCC is a little bit deeper, the beam is a bit more elongated, so you're actually hitting more tissue. And it looks like it does matter. So the bigger the focal volume, the more decrease in GABA we saw. So perhaps that explains what we found. Another possibility is state dependency. So this is just the default mode network at rest. Just a reminder that people were at rest during this task. So yeah, the ACC is not very active when you're mind wandering, while the PCT is very active. So perhaps this is what matters. And the other thing that maybe is possible is that there is a lot of individual differences in the ACC anatomical structure, but also even tissue composition, thinking about, so maybe that's also what happened, but we're still investigating those questions. All right, so I hope that I convinced you that it is possible to find effects after energy has been applied for a few minutes. And I hope that this is also, it is to me very, very exciting and very promising because it opens doors to so many therapeutic applications for those of us that are interested in patient groups. So thank you very much, particularly Titi and Tristan, who led most of this work, and to all my collaborators.
Okay, thank you, Elsa. So here I have questions from audience.
Hello. So he in the under said that there was a difference in the effect of 15 minutes of simulation 15 minutes. But did you maybe monitor online differences after 15 seconds? So
No.
Yeah, that's that question.
80 seconds is quite short, and you're kind of holding your breath because you're maintaining the transducer space. So the only thing I can tell is that the room was very silent for those 80 seconds and nothing was happening. But yeah, no, I can just say that they were in a state of rest when they were being submitted to them. But I didn't monitor any of them. We didn't monitor any of them.
So thank you for the talk. For this panelist. Perhaps I missed it. Did you talk about resting state continuity from the anterior cingulate even in the absence of changes in the MR spec signal?
What do you mean, in the absence of the QS?
Yeah, so even in the end, so even though there was no change in the spectroscopy in the anterior cingulate, changes. I'm sorry?
Yes. So we didn't see any difference between the baseline and the TOS, but obviously, the ACC is nicely coupled with a lot of different areas, and you can see it very clearly. It's just that when we were doing the differences between the baseline session and the TOS session, basically it was equally coupled with its effective network or resting state network, FUN1.
Okay.
Yeah, so that's what I was meaning by we didn't find anything, but obviously it is the fMRI was still good quality, so you could still see the covariation of the signal with the rest of the brain. Yeah.
Great.
Interesting talk. Do you have some data about direct vascular effects? If the ultrasound basically changes the vasculature in its reactivity, then all the effects we have seen, apart from the behavior, could be explained by changes in blood volume, changes in reactivity. So is there evidence that the ultrasound changes the tone, the vascular tone, its spontaneous modulation by respiration and so on. Is there some research on that?
Are you talking about the interface between the cell and the brain?
Or I think the most change can be explained by neurovascular, but the dominant one, which is the focus one, which see a change in connectivity when one area is no longer showing spontaneous changes the response. So I'm just saying that a neurovascular effect might drive a change of movement and change in GABA. So I just wanted to kind of ask if there's some research ongoing on direct vascular effects.
I mean, no, I think it's very interesting, yeah, and definitely need to be answered, but I don't know.
Thank you.
Yes.
Yes, thank you very much for the wonderful talk. So, I have a question regarding the last study that you talked about, and you briefly touched upon state dependency and how the agency maybe uses the government and consensus. So I was wondering what your thoughts are on that and how we can maybe still do something in our study design or change study design and research to enhance it.
Yeah, I think this is a very, very good question. So we thought about it as perhaps we can look at the default mode network and the strength of coupling between the PCC within that network and see if that relates to GABA change. So the idea was if people are very mind wandering, then maybe you see even more of a GABA effect. But it is a bit so it was a bit tricky because the GABA level and functional coupling, so the decrease in GABA and the increase in coupling was not that related in our study to start with. So we thought that it was a bit of an ill-defined way of looking at the data because of that. It was still going in the right direction, but it wasn't significant. But yeah, so I think this would be the way to do it. But also having studied that, particularly look at increasing some activities in some regions and make sure that we know when it will happen. So for example, pain model would be very interesting for state dependency. Also, a reminder that the non-human macaque model, so they were anesthetized, they were in the same state. So that's also maybe why the things could look different between the two.
Any other questions? No questions from Zoom? No? No? Okay. So thank you so much. Okay, so let's move on to the oral session. And as for the technical problems, so the order of the talk is changed. So Benjamin Kopp's talk is first, and then the video talk by Jae-Jun Zhu, and then Honsun Go talk will be. Okay, so the next speaker is Benjamin Kopp. The title is an auditory compound can drive online effects of transcranial ultrasonic stimulation. So please, Dan.
I'm not trying to get my presentation up here. Mika. Okay, hello everyone. My name is Benjamin Kopp. I'm a PhD student at the Dulles Institute in Dr. Leonard Verhagen's Cognitive Neuromodulation Lab. And today I'd like to tell you all a little bit about one of our studies, which I've quite ominously titled "An Auditory Confound Can Drive Direct Effects of Transcranial Ultrasonic Stimulation in Humans." Let's just start off with some background. I can't tell if it's, oh. Backgrounds. Yes, so as I'm sure all of you know, transcranial ultrasonic stimulation, or TUS, is a up-and-coming neuromodulatory technique for human applications, and it has tremendous potential for both clinical interventions and also for oh, I have to get my click button to work. No. Clinical interventions, and also for just normal research applications. And what's really, really important at this kind of fundamental stage and this infancy, essentially, of transcranial ultrasonic stimulation in humans is that we're sure that the neuromodulatory effects that we're observing following TUS can be attributed to direct neuromodulation as a result of TUS. And so this talk is going to overlap slightly with Talika's talk as well, so I'm going to apologize in advance if there's a bit too much overlap. So now we're finally in a place where we can non-invasively neuromodulate deep brain structures, open up causal investigations that were never before possible. But it's really important that we're accounting for direct neuromodulation here. And the reason why I'm bringing that up is because of some animal work and because of some human work. So here you can see some work by Guo and Sato. I apologize, I spelled Guo wrong here. And I'm going to draw your attention to the middle and lower panel there. What you can see is some calcium imaging, and the activation patterns following transcranial ultrasonic stimulation and stimulation with sound alone are very, very similar to one another. And on the right, you can see that there is an effect of ultrasound in these animals, but when they're deafened, that effect goes away. And we're seeing a relatively similar finding here in some human work. Here we see some EEG data, ERPs, and here ultrasound has been applied both with and without a masking stimulus. And then there's a sham condition where they've used a masking stimulus as well. And what you can see is that neural activation pattern is very similar, also indicating that it has its root in auditory peripheral stimulation. So this really highlights the substantial impact that these peripheral confounds, particularly auditory confounds, can have on common outcome measures of neuromodulation that we use in neuroscience. So I'm going to segue over to our own research, but I will get back to this later. So in our own study, just to give you some background, we were just setting up a lab. We're a relatively new lab in the Netherlands. And we thought, where do we start, right? And we chose the primary motor cortex, which is a quite popular target, really, for a number of reasons. And primarily because we can get a fairly straightforward and objective readout of motor excitability when targeting this area. And what we do there is we take a concurrent TUS TMS approach. So here you can see our combined system. And if I just expand this for you, then you can see that we keep ultrasound in line with the middle of the TMS transducer. And that allows us to apply stimulation of TUS and TMS at the same time. And what we know from work by Lagon and colleagues, for example, is that at least in the physical domain, these two are not interacting with one another. So the ultrasonic pressure wave is not affecting the magnetic field induced by TMS, and TMS is not really affecting, at least not significantly affecting, the pressure wave induced by TUS. Now, the general approach that we're taking here is quite simple. We apply TMS over the primary motor cortex, the hand nod specifically. And what we do is then we measure an MEP over the first dorsal interosseous, the FDI, and the amplitude of that MEP is thought to reflect motor cortical excitability. Now, when we apply ultrasound at the same time, we expect to observe a difference in the amplitude of the MEP, and that difference would reflect a modulation of motor cortical excitability induced by TUS. So that's the general approach that we take, and it's not original. It's been done before. So here you can see some work by Professor Lagon, Dr. Flamenco, and then Dr. Shia. And what we've observed here is direct online motor cortical inhibition following ultrasound, which is reflected by an attenuation of MEP amplitudes following TUS. right? And what you can see on the lower panel is that this is a transient effect. So on the lower panel, they applied ultrasound for 500 milliseconds. And you can see that motor inhibition does not exceed stimulation for longer than about 20 milliseconds. Now, FUN22 also did a great and a novel investigation into the substantial impact that different stimulation parameters can have on the neuromodulatory outcome of TUS. Here I've highlighted only one of them, but they did a lot of parameters. And what they observed here is a stimulus duration dependent effect of TUS, where only longer stimulus durations of 400 milliseconds or longer, could significantly attenuate MEP amplitude. So, in our own lab, we set out to do a bit of a replication and an expansion on this work to kind of jumpstart TUS in our own lab. So here we're going for online motor inhibition, and we've also included a kind of cut up version of that stimulus duration dependent effect. We're applying TUS at 100 and 500 millisecond stimulus durations. Our hypothesis there is that 100 will not significantly attenuate MEP amplitude, while 500 milliseconds will. Then we also decided to conduct a novel investigation. I think we made that decision just alongside Talika at the same time, to also look at stimulation intensity. So we applied stimulation intensities, spatial peak pulse average, at around 32.5 and 65 watts per centimeter squared. Now, the stimulation parameters that you use are pretty straightforward, quite common ones. We're applying, like I said, stimulus durations of 100 and 500 milliseconds with a trial duration of around 6 seconds. And if we zoom in here, then what we can see is we're using a 30% duty cycle. So we have 0.3 millisecond long pulses with a 1 millisecond pulse repetition period. And something that's really important to note here is that specifically this protocol has a very, very present auditory content. It sounds like a very loud beep. You're never going to miss it. It is really, 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 really loud. That's just something that I want to highlight here. Not every protocol is associated with an auditory confound that's as prominent as this. So because we have such a strong auditory confound, moving back to those effects of peripheral stimulation that we saw from both animal and human work, we wanted to make sure that we really tightly controlled for that confound. And so what we've done here is included 2 control conditions. We applied on the left, you can see we've just done on-target TUS, so ultrasound is applied to the primary motor cortex, the hand knob over the left hemisphere. But we also apply ultrasound over the contralateral face representation for a number of reasons. There's a poster later, maybe questions if you want to get into the details there. So that condition not only controls for the spatial specificity of ultrasonic neuromodulation, but it also quite tightly controls for the experience of the auditory confound. And then finally, we also included a sham condition where no stimulation, ultrasonic stimulation, is applied at all, but we administer a sound over bone conducting headphones, and that sound was made to sound exactly the same in our experience as the actual auditory confound associated with our transducer at 1000 hertz. To provide just a little bit more detail here, so we're doing 100 or 500 milliseconds. We apply TMS 10 milliseconds prior to offset, which is relatively standard in this paradigm as well. And we've also included a baseline condition. I do want to highlight, it's possible that there would be audible differences between stimulation that's applied over the hand area in the left hemisphere and over the face area in the right hemisphere. Those could sound different, right? Because the sound is primarily through bone conduction, and those are different segments of the skull. To account for any potentially audible differences between those stimulation conditions, we applied them both with and without an auditory masking stimulus. That auditory masking stimulus was identical to the stimulus that's used for sham stimulation. In this sample, the sample is 14 participants. We applied the stimulus over bone conducting headphones, and because they're Bluetooth, we had a 100 millisecond extra padding needed so that the timing would be good enough. So here, that masking stimulus starts around 50 milliseconds before ultrasound starts and ends around 50 milliseconds after. And we did improve on that later, which I think I'm getting to now. So we ran a second experiment, second sample, we pre-registered it with 32 participants. And in this sample, we decided to only look at the effective stimulus duration of 500 milliseconds. And again, we're looking at 2 different intensities, this time 10 and 30 watts per centimeter squared with a 250 kilohertz transducer. We're using the same control conditions and we're applying everything both with and without a masking stimulus. Here we made a number of technical improvements as well. And one of those technical improvements was making sure that the auditory masking stimulus / sham was exactly timed the same as ultrasound. So I want to move over to our results. Happily, we were able to replicate this core robust finding of online motor inhibition following ultrasound. On the left, you can see our first sample of 12 participants that were included. And on the right, you can see our sample of 28 participants that were included in the final analyses. So this is great. We were happy. And then we were a little bit less happy because what we observed was a non-specific effect of transcranial ultrasonic stimulation. So here you can see baseline in gray, TUS in dark green showed a significant reduction from baseline, but stimulation of a control condition, so stimulation of the face area, also resulted in this MEP attenuation. On top of that, administration of a sound alone, regardless of whether we did it over bone conducting headphones or whether we did it in a very precisely timed manner, also resulted in this inhibition. So what it looks like is that sound, rather than ultrasound, might be responsible for the motor inhibition that we've observed thus far. And it's going to get worse. Here we go. We also looked at stimulus duration, and we replicated the stimulus duration dependent effect of ultrasound, where if I draw your attention to the left panel, you can see that 100 milliseconds does not significantly, you'll have to take my word for it. It does not significantly change from baseline, whereas 500 does. But again, we see that same pattern for both the stimulation of a control region and for administration of sound alone. This is from the first sample. So if anybody's wondering why the sham looks a little bit less fancy, I think it might have to do with the timing. So here we have kind of a little bit more evidence that it's sound and the duration of sounds as opposed to direct neuromodulation. Now, we also looked at stimulus intensity. Here you can see, again, the first sample, so 32.5 and 65 watts per centimeter squared, and we did not observe a significant difference in MEP attenuation from baseline in those two conditions. And what we've also done is, with the wonderful help of Andre Chetverikov, who is here today, we ran simulations of acoustic wave propagation, and we double-checked, or simulated at least, the intracranial intensities. And here you can only see this is not enough for an actual regression, but we don't see any kind of strong relationship between the simulated intracranial actualized intensities and MEP reduction from baseline. Simulations for the second experiment, we're still working on them, so that's why I can't show them here, but I can show the rest of what we have for the second experiment. So this is all of the data, all together in one place. Very sad, flat line for us today. So we have baseline, we observe a reduction for both 10 and 30 watts. None of these conditions significantly differ from one another. They are all very similar. When we applied a masking stimulus in the desperate hope that maybe we would still kind of find something, you can see that that doesn't make any difference either. Again, sham reduces from baseline. And what you also can see on the right here is an assessment that we ran of our masking stimuli. So we obviously applied these masking stimuli, but it's important to be able to tell whether participants are actually blinded by them or not. So that's what you can see on the top here, where masked stimulation looks like blinding is working pretty well, whereas unmasked, not so good. But just because participants can't tell whether or not they've received ultrasound does not necessarily mean that they can't hear differences anyway. So what we've also done is presented people with the same stimuli twice, so hand area, or two different ones, so hand area, face area, with a masking stimulus. And what we see here is that while I still rate them as similar when there's two different sounds, they can hear more of a difference when there's different stimulation sites than not. I'm going to wrap up. So the take-home messages, again, this is about a stimulation protocol with a very, very prominent auditory confound. And that auditory confound reaches your cochlea via a number of pathways. One is via air conduction for a protocol this loud, and primarily it goes through shear waves and bone conduction, which I think Talika talked about as well. So just a little bit on future perspectives. What can we really do about this? I think it's a very important consideration now that we know that these confounds can affect now also an extra outcome measure of neuromodulation that's used relatively often, tight control conditions that make sure that we're controlling for the auditory confound and also potentially somatosensory confounds are incredibly important. I think masking will also be very important in the future, and perhaps titrated masking will be required. An important consideration there is also what way you're going to do masking, whether that's bone conducting headphones, earphones, speakers. There's of course always a future in alternative protocols, offline protocols, inaudible protocols. And then of course we also have ramping, which can make protocols that are slightly audible, inaudible, and may make audible protocols less audible. So I think that's it for today. I'm happy to take any questions. I want to give a really big thank you to my whole lab, especially to Judith Lefkis, who is an integral part of this project, Marlon Engels, who is here with us today, who is also an integral part, and Brittany from Bake, who helped us a lot with data acquisition as well. We are looking for a postdoc, so please email Lennart if you want one. And I think that's it. Thank you.
Thank you very much. Questions from the audience?
So you microphone.
And if you think about what is the mechanism, is it the kind of a starline thing? Is it something that goes through the reference system? Is it translating? So how do you look a little bit personalize it now the phenomenon is there and how you create it?
Okay. Sure, yeah. So the question is, I think, two parts, if I got it correctly. The first is, did we look at the temporal scale of these effects? And the second question is, what we think the potential mechanism of inhibition is. So for the first one, no, we have not looked at the temporal scale, but we would love to, and that's definitely something that we are considering looking at next. So obviously you could consider timing the pulses, kind of like Shia et al. Did before and later on multiple trials to try to like delineate what the time course of inhibition is. That approach would also, I think, help with coming up with what the potential mechanisms are. As for the mechanisms, I don't want to speculate too much, so I'm just going to name a couple of things. I think startle is possible. I think it's possible that you're kind of in a motor deprived state, and just like in other kind of, for example, if you're in a very dark room and you hear a sound, sometimes you can see something, sometimes you can feel something. In that way, you're sitting still and you hear something. Maybe that is then in a multimodal way connected to the motor system, but this is definitely not my expertise. I'd love to hear your thoughts about it because I have to write up my discussion and I would love to, to hear what you think.
Yeah, on a similar way, your data works for three conditions very similar from inhibition. What three groups have in common is that they receive a slow frequency TMS, which is inhibitory. You consider whether that could be a potential inhibitory effect on the motor over time.
You know, I think there's so much that we don't know. And no, I don't. I'd like to talk to you after, but no, sorry, I don't have any thoughts.
Any questions? No questions from the Zoom? No? Okay, thank you very much. So the next speaker will participate online. So that's next speaker is Guo. So the title is ultrasonic neuromodulation produces amplitude-dependent off-target cortical activation in awake transgenically dependent mice.
The speaker is available online for questions at the end of the webinar.
Hello everyone, my name is Hongsen Guo. I'm a postdoc in Professor Mikhail Shapir's lab at Caltech. Today I'm going to present ultrasonic neuromodulation produces amplitude-dependent off-target cortical activation in awake transgenically deafened mice. First, I would like to review the background of the auditory confounds during ultrasonic neuromodulation in rodents. Then I'm going to talk about why and how we created a transgenically deafened mouse model for ultrasonic neuromodulation. And last, I would like to share some results we got using this deafened mouse model, including elimination of auditory confounds and residual non-auditory off-target brain activation. And also we saw decreased fluorescence signal at the ultrasound focus and localized and propagated brain activation with high-intensity ultrasound. A few years ago, Tom and I reported ultrasound can cause extensive brain activation in rodents. In Tom's study, he simultaneously stimulated mouse visual cortex and recorded the whole brain using wide-field calcium imaging. He found that the ultrasound did not activate its focus, which is the visual cortex. Instead, ultrasound causes activation of the auditory and somatosensory regions bilaterally. And I got the same results using guinea pig as a model. And I found ultrasound can activate the auditory and somatosensory regions of guinea pigs, and this activation can be successfully eliminated by surgically deafening the animal. And these findings suggest that the ultrasound did not activate the brain, and it can only indirectly cause the brain activation by activating peripheral auditory pathways. So therefore, we may want to investigate the ultrasound neuromodulation after deafening the animal. However, there are a few limitations of the method that Tom and I used in our studies. For example, in my study, I transected the auditory nerves and broke the cochlea. However, they are very invasive to the animal, and it's not ideal for weak and survival experiments. And in Tom's study, he used kanamycin and furosemide to deafen the animal, and it can cause systemic toxicity to the animal, and also the hearing loss is incomplete. To overcome these limitations, we use heterozygous Pro4F3 DTI mouse as our animal model. In this mice, gene encoding the human diphtheria toxin receptor was placed downstream of the Pro4F3 promoter. Since mice are 10,000 times more resistant to diphtheria toxin than humans, the injection of diphtheria toxin will achieve selective ablation of hair cells without causing side effects and systemic toxicity to the body. More importantly, by breeding with GCAMP mice, we can get double transgenic mouse for calcium imaging. The protocol is pretty simple. The mice will receive 2 doses of diphtheria toxin injection, spaced 2 days apart. The mice will be completely deafened after 2 weeks. To confirm the double transgenic mouse are truly deafened after the administration of diphtheria toxin, we examined the cortical responses to sham, sound, and flashing LED lights in head-fixed, fully awake mice. There were 3 groups of mice being investigated, which were C1DT, the C1 GCAMP mice injected with diphtheria toxin, PO-C1 saline, the double transgenic mouse injected with saline, and PO-C1DT, double transgenic mice injected with diphtheria toxin. And indeed, you can see a representative Example from each group. It's clear that sound causes auditory responses for C1 DT and for C1 setting groups, but not for C1 DT groups. Further statistical analysis in E suggested that auditory responses in pole C1 DT were eliminated while the visual responses to flashing LED lights were not affected, suggesting that the toxin have successfully deafened the mice without causing any damage to the visual system. We then investigated if the ultrasound-elicted auditory confounds can also be eliminated in the deafened mice. We first tried a low center frequency 270 kHz transducer and applied continuous ultrasound for 400 milliseconds to the visual cortex, and we found that the auditory confounds were significantly reduced but not fully eliminated. At high pressure above 500 kPa, we still observed activation of bilateral somatosensory and visual cortex. And this off-target activation may be other types of sensory confounds, such as skin and retina activation by propagating ultrasound waves. Here are some statistical analyses between the normal hearing mice group and the deafened mouse group. And from A, the average visual responses to light were similar between the two groups. And we also compared the ipsilateral visual cortex at the focus with contralateral visual responses and found they are not significantly different from each other, suggesting that ultrasound didn't cause localized activation when using the 270 kHz transducer, and the activation at the focus were non-auditory sensory confounds. Since the 270 Hz transducer still caused sensory confounds in deafened mice, we switched to a 916 Hz transducer, which has a much smaller focus. We found that the sensory confounds were successfully eliminated. However, we didn't observe any direct activation with ultrasound. Moreover, when further increasing the pressure to 1 megapascal, we started seeing the decrease of fluorescent signal instead of evoked calcium responses. We were not sure if this decrease of calcium responses and the negative ultrasound brain activation results were due to the distortion of ultrasound waves through the skull. So instead of delivering ultrasound through the clear skull, we developed a TPX window setup where the bilateral skull covering the anterior motor cortex to the posterior visual cortex was replaced with two pieces of TPX plastic, which is an optical and acoustically transparent material. We delivered ultrasound through the TPX window to the visual cortex with different ultrasound pressures, ranging from 100 kPa to 1 megapascal. However, we didn't observe any direct activation at the focus. Instead, we observed the decrease of fluorescent signal. After measuring the temperature during the sonication, and especially after comparing our results with Dr. Estrada's amazing ultrasound neuromodulation study published in Science Advances last year, we concluded that we reproduced what had been reported in his study, that the decrease of fluorescence was not due to neuronal inhibition, but was due to the fluoriphore thermodependency. When the pressure being used is equal to or greater than 1.6 megapascal, we observed strong calcium responses at the ultrasound focus, which subsequently propagated all over the ipsilateral hemisphere, and those very strong responses closely resembled to responses of focal cortical seizures, suggesting the local inhibitory circuits might be severely disrupted by high-pressure ultrasound. And these results are also consistent with Dr. Estrada's Science Advances study. And that's all. And I would like to thank my advisor, Professor Mikhail Shapiro, and my former advisor, Professor Doris Cao, who have been supervising this project and giving me valuable advice. And I also would like to thank many of the lab mates who helped me on this project, and also the NIT Brain Initiative. Thank you all. Please let me know if you have any questions.
Hello, hello. Can you hear me? He seems to be on the our microphone is working. Yeah. So yeah, I'm sorry for the technical problems. There is technical problems, so we cannot talk to simultaneously. So then, if you have any questions, so please send the email or post the questions on YouTube later. Okay, thanks. So then I'll move on to the next talk. Thank you very much, Dr. Guo. Okay, the last speaker of this session is by participating in online, and this is we displayed a pre-recorded presentation. Okay.
Hello everyone, I'm Zhu Jiejun from the Hong Kong Polytechnic University and Guangdong Institute of Intelligent Science and Technology, China. I'm very happy to be here sharing with you our recent findings that the piezo-1 contributes to ultrasound neuromodulation in vivo. So, ultrasound neuromodulation. I believe most of us here would appreciate the great potential of ultrasound in the application of neuroscience due to the widely observed bio-effect in multiple animal models. But we're also aware its limited effectiveness, which may partially due to our incomplete understanding of its molecular mechanism. We know that the ultrasound could generate heat, cavitation, and acoustic radiation force while propagating its energy, but the acoustic radiation force has become a consensus more recently, especially with the finding that the neurons could operate the mechanical transduction at the with the force at the low piconewton level. More importantly, in vitro, accumulated evidence has demonstrated various mechanosensitive ion channels contributes to the ultrasound neuromodulation, although the in vivo evidence are still limited. And in our lab and Mary Maduk's lab, we both found that the piezo-1, which is well-established mammalian mechanosensitive ion channels, mediates the ultrasound neuromodulation in vitro. And since the piezo-1 is the upstream of various mechanotransduction pathway, we were wondering if the piezo-1 is also plays an essential role in ultrasound neuromodulation in vivo. What we do here is that we first generate a piezo-1 conditional knockout mouse model through Cre-LoxP system. We use this piezo-1 LoxP mouse line generated by Adam Petterpolian's lab, and we can see here that the axons here are flanked by LoxP sites in this mouse strain. And with the recombinase expression in targeted neurons, these strains can then be deleted. And after that, the calcium signaling, the mouse behavior, the electrophysiology, the molecular changes is then tested under the ultrasound stimulation so that the piezo-1's role in ultrasound neuromodulation can be studied. So here's our results. We first test the piezo-1's expression in the mouse brain with IF staining. Firstly, we test the piezo-1's antibody specificity. We can see here that several neurons would be stained with piezo-1, but this signal would be abolished with the antibody's pre-incubation with its blocking peptide, which indicates the antibody's specificity, and we thus use it for our experiment. And staining with the brain slice, we do find that the piezo-1 is indeed expressed in the mouse brain, and its co-staining with MAP2 further indicates its expression in neurons. And staining with the brain slice of our later generated P1KL mouse model, which we can see here that lower signaling is found, and thus this data further indicates the antibody's specification, and piezo-1 is indeed expressed in the mouse brain. And what makes things more interesting is that while we do the staining in the brain slice, we find piezo-1 is higher expressed in specific brain regions, including the CEA, BNST, PVN, EW, RN, etc. And we take the cortex and CEA for our further demonstrations here. Testing the inward current induced by user 1, which is a piezo-1's agonist, we further confirm the functioning of piezo-1 in the mouse brain. And recording the calcium signaling by fiber photometry, we can see a well synchronized of calcium signaling induced by ultrasound stimulation. And if we compare this signaling with CEA and cortex area, we can see a higher acoustic pressure dependency of CEA neurons comparing with the cortex neurons. And if you do remember that in the previous slides, we report our finding that a higher piezo-1 expression in the CEA areas. So together, this result support our hypothesis that the piezo-1 may play an important role in ultrasonic neuromodulation in vivo. But obviously, more demonstration is required. And we take the motor behavior as our demonstration. As some of you may be familiar with a phenomenon that when the ultrasound is stimulated, say the right motor cortex area, a mouse left limb movement could be triggered. So let's see how it looks like. We can see that when the ultrasound is on, the light indicates the ultrasound stimuli. When the ultrasound is on, the left limb movement is triggered. But if we express the recombinants in the P1LOXP mouse line, which means that we knock down the piezo-1's expression in neurons in this brain area, this phenomenon is significantly reduced. We can see that the light did not trigger the left hind limb movement. And if we stimulate this mice on its left motor cortex area, where the piezo-1 is still remains, its right limb movement is there, can be triggered. So let's see another one. So together our results demonstrates that the piezo-1 is indeed plays a role in ultrasonic neuromodulation in vivo. To more quantify this motor response, the EMG signaling was then recorded. And here's the results and the statistic results. And we can see here that in the control mice, the EMG signaling can be induced by the ultrasound in an acoustic intensity-dependent manner, while the P1KO mouse showed significantly lower EMG signalings here, demonstrated that the piezo-1 should contribute to the ultrasound-induced motor behaviors here. And more directly measured the neuronal activity induced by the ultrasound, the fiber photometry is then adopted. And we can see here that with the piezo-1's knockdown, the calcium influx induced by the ultrasound is much lowered. And here's the statistic results. So together, this data indicates that piezo-1 should contributes to the ultrasound-induced neuronal activity in vivo. And C-force expression, which is a standard neuronal activity marker, also showed consistent results. We can see here that with the piezo-1's knockdown, the number of activated neurons by the ultrasound stimulation is much lowered. And here's the statistic results. So, taken together, our findings demonstrates that piezo-1 is functionally expressed in the mouse brain and also plays an important role in ultrasound neuromodulation in vivo. And since piezo-1 is found specific distributed in some brain area, if I may say, a stress-regulating brain area, it thus may also act as a target for studying and treating psychiatric diseases. Thank you.
The chat so please email us. Okay, so that's the this is the end of the oral session. And so we have a coffee break now, and the next session will start the 55. 3 o'clock. Okay. The next session will start at 3 o'clock. Okay, so let's have a coffee break. Thank you very much.
有人提问？不是，已经 Thank you very much 了。我确实听不到他在说什么。哦。
You are now online.
Hello. Yes, I'm online, but I think I cannot hear you.
Okay. Can you hear me?
You know, now and then it's just
I'm sorry, I missed you. So I missed your presence online. So if you have any questions from audience, so please ask a question. Do you have any questions? No?
Okay.
Wait a minute. So the your question is the piezo the frequency dependency of the piezo response. Do you have any observation about that?
Sorry, I cannot hear your question.
So her question is, can you hear me?
Now and then, but not very clear. Okay, how about email me?
I'm sorry, there is a technical problem, so we cannot communicate simultaneously. So if you have any questions from the audience or from the Zoom participants, so please post the questions via email or the chat from the chat. Okay, I'm sorry for the trouble. So thank you very much for Dr. Zhu.
Thank you.
Got something for the industry, okay? Yeah. So we're gonna the mic is still on, I guess. I think the mic is still on.
Hello everyone, I'm Zhu Jie.
Hello everyone, this is Liming Chen. The title of my presentation is transcranial focused ultrasound suppresses thalamic heat pain response and reorganizes effective and resting state functional connectivity networks. I'm interested in study pain because it is the leading cause of disability and opioid addiction. For many years, pain has been viewed as a sensation that is similar to vision or touch. However, since the discovery of the functional bolt MRI contrast, now the investigators can visualize and monitor brain response when subject or pain patient experiencing pain inside the magnet. Hello. As the key hubs within the PAN network. In our study, we focused on 3 brain regions: the anterior cingulate. Hello everyone, this is Liming Chen. The title of my presentation is Transcranial Focused Ultrasound Suppresses
it again. So please return to your seat. Hi, hello. So we'll start again. Now. Okay, so thank you for coming back. So the last oral sessions. So the talk by an invited speaker. So I'm so pleased to have Dr. Liming Chen from Vanderbilt University as an invited speaker of the FUN22. And then last session of the neuroscience, today's neuroscience session. And unfortunately, she cannot attend physically in this this meeting. So she kindly provided us a pre-recorded talk. And is she's connected? Okay, so now she seems to be connected. So after displaying this video, she will kindly answer the questions from the audience or the participants from Zoom online. Okay, so now we'd like to start the pre-recorded talk.
Dynamic heat pan response and reorganizes effect. Hello everyone, this is Liming Chen. The title of my presentation is Transcranial Focused Ultrasound Suppresses Thalamic Heat Pan Response and Reorganizes Effective and Resting State Functional Connectivity Networks. I'm interested in studying pain because it is the leading cause of disability and opioid addiction. For many years, pain has been viewed as a sensation that is similar to vision or touch. However, since the discovery of the functional bolt MRI contrast, now the investigators can visualize and monitor brain response when subject or pain patient experiencing pain inside the magnet. These studies have revolutionized our understanding about pain and pain mechanisms, and the current hypothesis is that pain actually emerged from a coordinated activity of integrated brain networks. And this observation have led to the people try to come up with neuromodulation tools that can perturbate the network and then reach a goal of pain relief. These slides show you the brain regions are considered as the key hubs within the PAN network. In our study, we focused on 3 brain regions: the anterior cingulate cortex, the thalamus, and the PAG. We choose these 3 regions because this area has been the target for deep brain stimulation for pain relief. And in this presentation, I'm going to show you most of the data from ultrasound stimulation of the thalamus and one data set from the PVG stimulation. The ultimate goal of our project is to develop an MI-guided transcranial focused ultrasound system with real-time feedback that can be used to stimulate pain-related brain regions and networks for pain relief. The objectives of the project is to first demonstrate that moderate intensity focused ultrasound can suppress painful heat evoked response in the targeted thalamic ventral posterior lateral nucleus. This nucleus is involved in pain processing in the human and animal brain. The second objective is to investigate ultrasound's effect on the thalamus's effective pain network and resting state pain network in a non-human primate model. In our approach, we combined simultaneous MI with ultrasound stimulation to allow us to monitor ultrasound's modulation effect. We also integrated the real-time MI acoustic radiation pulse imaging, RF image, to provide a real-time feedback about the accuracy of the ultrasound targeting. This slide shows you the methodology details we used in our study. We used 128-channel ultrasound array, and this array provides a very focal stimulation of the target. We adapted the ultrasound parameter from our previous studies that looking at ultrasound modulation of the tactile system. Our previous study have shown that high intensity ultrasound can excite brain activity of brain neurons, while moderate intensity ultrasound can suppress brain response. And in this particular project, we implemented a 650 kilohertz ultrasound pulse and delivered at 550 kilopascal pressure intensity. And this image shows you how we identify the VPL nuclear using functional imaging at each individual subject level. The purpose is to customize the targeting in each subject. Before ultrasound stimulation, we acquired functional imaging data when deliver painful heat stimulation on the hand. So here on the top three image, you can show the VPL nuclear showed robust response to the painful stimulation. And this region is corresponding to the VPL nuclear illustrated in the histological slices and also the atlas. Once we identified the target using functional MI, and then we will guide the placement of the ultrasound targeting. The bottom three images are the RF image we acquired, and you can see the ultrasound beam was successfully targeted at the VPL nuclear, where we see robust functional response. This approach allows us to identify precisely the brain region showing the response, and then we can make sure our ultrasound stimulation is actually applied at this region. This arrow just shows you the correspondence between the VPL activation and MI image and also an anatomical atlas. This imaging shows you our data acquisition strategy. Each imaging session lasts about 6 to 7 hours. Animal were lightly sedated. Once the animal is sedated, we put the animal into the static frame and insert it in the MRI magnet. First of all, we acquire high-resolution T1 weighted structure image and RF image. The T1 image will be used to align the functional imaging from each session to the monkey atlas for group level analysis. The RF image will ensure our ultrasound beams reach the target as we intended. Before ultrasound neuromodulation studies, we acquire resting state functional MRI data before and after. This design allows us to look at the post-ultrasound exposure effect on resting state network. And during functional MRI stimulation, during the ultrasound stimulation, we acquire functional MRI data and in runs. Each run is about 15 minutes long. Within each run, we alternating two experimental conditions. One is the heat of the hand alone, and the second is heat hand plus fast stimulation of the thalamus. These two conditions alternated, and this design allows us to precisely compare the response between these two experimental conditions. By the end of each imaging session, we also acquire safety MRI data to ensure the ultrasound exposure did not introduce structure disturbance. The imaging data are acquired with a pair of flat surface coil. The heat stimulation is 47 degrees C applied on the monkey's head. This temperature will elicit heat painful sensation on humans. The whole brain functional MRI data are analyzed in MATLAB and FSL using macaque monkey atlas. The functional data acquired at high resolution of 1.43 by 1.3 by 3 millimeter cubic size. All the quantification are done at the group level. I'm going to show you the results from 3 different angles. First is ultrasound modulation of painful stimulation evoked bold response at the target and off-target regions. The second is ultrasound modulation of effective connectivity of the thalamus PEN network. The third data set is ultrasound modulation of PEN resting state network. This image shows you the whole brain response to the painful heat stimulation. These activation maps are similar to what has been reported in the humans and animals during the painful stimulation. And the area is widespread as we expected. The right image shows you the thalamus response was suppressed by ultrasound. You probably can appreciate the overall number of activation is smaller, and some areas activation are suppressed and are not detected. For example, if you look at this image, we have 1, 2, 3, 4 activations in the cortex, and when the thalamus response was suppressed, there was only 1 cluster remained. To better visualize the effect, we plotted the activation on the inflated brain. So the left is the response to painful stimulation, and you can see many of the areas are illustrated in this core PEN network. After the fast suppression of thalamus response, the overall response, the number of activation reduced. For example, the prefrontal area, you can see the activation disappeared. And some of the areas in the temporal lobes also reduced. We next quantified the extent of the suppression using ROI-based time course analysis. And here you can see at the target VPL, the heat response is about 0.5%. When the moderate fuzz was delivered, the heat response was significantly suppressed. The other 3 areas are the interconnected PEN regions to the thalamus. They all showing suppression when the ultrasound was delivered at the thalamus. So this data indicate when the thalamus pain response was suppressed, the interconnected areas were also affected and also showed suppressed response. This plot, the bar plot, showing the peak differences between these two experimental conditions. And you can see they're all quite significant. We also quantify the time to peak measurements between these two conditions. With the presence of fast stimulation of the thalamus, the time to peak varied or changed at each individual brain region. Some of them gets shorter and some of them gets longer. And we don't know how to interpret these findings yet. The next set of data, we also asked the question when the thalamus painful response was suppressed and how this suppression affect the thalamus network. What we did is we performed analysis called psychophysical interaction analysis. And this analysis basically extract the heat stimulation evoked both response from the thalamus. And here is the response from this region, where the blue time course. After we extract the time course, and then we asked what other areas in the brain show the very similar response curve during the stimulation. Based on the time course, so here you can see the blue curve is the time course during heat stimulation. And the blue blocks, that's when the heat and fast stimulation was delivered. And the red curve is the PPI or the regressor. So using this regressor, we can plot where in the brain show the very similar time course change as the seat of the thalamus. This analysis allows us to look at when the thalamus response was suppressed, how thalamus effective connectivity network change. After we extract the time to see how the it change. So it change? So we did, and how does it change? So we did two types of analysis. First of all, we performed the hierarchical cluster analysis to see how the thalamus painful network reorganize. And in the painful stimulus condition alone, you can see the thalamus pain network is organized in two big networks that we call it blue network and the red network. And then within the network, you can see the hierarchical organization or the connectivity strength between different regions. For example, this is at the sensory cortex, S1 cortex. And, and after the ultrasound suppressed the thalamus response, and you can see the organization of the network changed. And some of the areas has become more closely connected when the other areas connection gets disrupted or gets weaker. So this data showing when the thalamus painful response was suppressed, thalamus's connectivity to its region also changed. Some of the areas gets closer together, and some areas gets more far apart, or they have less similarity in the clustering analysis. The next analysis, we quantified the change of their connectivity strengths. And this matrix plot showing you the intensity of the connection by the color. The hotter the color, the stronger the connectivity is. So you can see during the heat stimulus condition, thalamus has a very strong connectivity to many brain regions. See this star region that's also showing very significant strong connectivity. When the thalamus painful response was suppressed by FUS, and you can see the overall connectivity strength was significantly reduced. Even some of the areas, some of the pairs, the connectivity may be strengthened. When we did time course analysis, the connectivity analysis from the subset of the brain region, you can see with the presence of the ultrasound at the thalamus, the connectivity strength significantly reduced pretty much across all the areas we selected here. That means the data indicate when the thalamus, the painful response was suppressed, the connectivity between the thalamus and its interconnected regions also suppressed or disrupted. And in the last data sets, we also look at how ultrasound suppression of the thalamus pain response affect the resting state network doing for the pain processing. So basically, we identified the seed showed response to painful stimulation and look at their connectivity during the resting state acquisition. And we compared the connectivity, the pairwise connectivity between and after ultrasound exposure. And the middle matrix plot, you can see, before the ultrasound exposure, the many brain regions are highly interconnected. After the ultrasound exposure and most of the activation, you can see it's decreased. The overall intensity is it's cooler in cooler temperature. Even some of the areas, the activity maybe increases, like this two, or this one regions. When we put all the inter-pair connectivity together, and regardless of each individual area, and showing here, that is the overall R-square value of the inter-area connectivity strength. The blue bar indicate before the ultrasound stimulation, and the red is after ultrasound stimulation. So you can see after ultrasound stimulation, the overall connectivity strength is significantly weaker. Whether it is the thalamus was suppressed or the VAG or VPG nucleus was stimulated. So this data, again, showing one of the brain regions, like thalamus, all the PVG was perturbated by suppressive ultrasound pulses, the entire PAN network change, the organization of the PAN network, and also the inter-area connectivity strengths, and all become weaker.
Okay, some audio data region. So, okay, so Dr. Lin Chen is now connected. Can you hear me? Dr.
Yes.
Chen? Okay, so I'd like to, so if you have any questions, so please raise your hand, and so she will answer the questions. So question from the audience. Yeah.
My question is, what is this the ultrasound alone condition? Has there been the ultrasound alone condition? There's been pain and ultrasound alone?
So your question is the ultrasound without pain means what is the meaning of the ultrasound without pain?
How can it's a fraction of those? Would be interesting to know what ultrasound alone has.
So what happens? So if the ultrasound is only the ultrasound is applied to the VDL.
Yes. So what we did collect the data. So when we collect the data, we have two different experiment conditions. The one I just showed was heat and heat plus ultrasound, and we also have heat fast alone stimulus condition. So to your question, we did that control, and some of the areas are overlapping, some of the areas are not overlapping, and yeah, so yeah, that's the overall conclusion. And when we stimulate the thalamus VPL nuclear alone, and some of the areas overlapping with heat response, and some of our areas are extra activations, not part of the PEN network. And we're quantify that. I don't have the data. I didn't present the data here, but we did. So the finding, yeah, I would just say some of overlap, some of don't.
Okay. So any other questions? So I have a question. So you show the resting set functional conductivity before application of the ultrasound and also the after the ultrasound application. And so during the simulation of the ultrasound, did you observe any accumulation effect during the acquisition of the functional MRI?
I'm sorry. So, what is your question? Whether we observed any network change during the first stimulation?
Yes. That's during the ultrasound simulation.
Yes. So, I mean, this is a very interesting question. So we're still investigating, because as you can see, when we acquire the first stimulation data, we usually acquire multiple runs. So we usually have 3 or 4 runs. That means we have about an hour apart. The duration is about an hour. We're still analyzing, and I don't have the results yet. The first thing we look at it was before and after. It seems that gives us a longer time delay, because we're not sure how long the ultrasound effect lasts. So that's the very first test we have done so far. And we do see a very robust modulation of the resting state network. So, and it's very possible we maybe have the power to see the change during the first stimulation, as you just mentioned. We have the data, we might be able to, but I don't have the answer yet.
Okay, thanks. Any other questions? Questions from the team? The participants? No? No. Any questions? Okay, thank you so much.
Thank you. I apologize for the brief interrupt of the video.
So I'd like to switch to and okay, so the industry pitches. Okay.
Hey people, wonderful to be here. Again, it's an in-person FUN conference. And I wanted to draw your attention to some of the special things that I now, as a member of the audience, rather than as an organizer, have been able to see. And it's such a real pleasure to see how physics and engineering, how basic fundamental neuroscience, how clinical work and cognitive neuroscience, so all of these domains are coming together to address a singular problem, or even maybe a singular challenge and puzzle. And I feel very bold and strengthened by some of the exciting results we've seen earlier today. And I feel also at what a luxury we are at this early stage of promising new technology, where we can rely on insight from so many different domains, where we can work together and we can really stand on each other's shoulders this time doing that. But I want to highlight that we have another unique opportunity here in the field, namely solving all of these challenges requires a very close collaboration between researchers and clinicians, and lastly, between industry. It's us for researchers who have come out often with crazy ideas and the stuff that we want to have realized on what we want to run, and we're also encountering new unexpected issues and opportunities. And I'm so happy to see some of the industry leaders in the field actually joining for this conference, allowing us to directly interact with them, to ask all the questions. And already early today, some people have put their hands in water tanks and felt what ultrasound was like. These are some truly unique opportunities to interact with industry. And that's also partly why we have now a series of industry pitches, where I'm going to ask some of them who've also generously supported this conference to introduce some of the work they do and how we can best interact with them. All of them are present. Actually, most of them are present. We also have a pitch from the FAST Foundation. They're not here physically, but with their setups where you can explore what they're doing, where you can ask all your questions and you can give all of their feedback, and really, again, as a whole field, work together. So it's my honor actually to start introducing the first of these pitches. They're ordered along in the sponsorship contributions. And I would like to ask if Brainbox and Sonic Concepts are ready. I can see Dan. Okay, fantastic. Yeah, come over. Floor is yours.
Thanks very much, Lennart, and to the organizing committee and to iTrust in general and FUN. It's a pleasure to be here and to be supporting the conference again and to see how it's grown and developed over the last few years, despite all the challenges we've all had to face. So for those of you, hopefully actually a lot of you in the audience here physically and online, hopefully know who Brainbox is, who we are, what we do. But don't worry, if you don't know, I'll hopefully kind of fill in those blanks for you. So we're mainly focused on non-invasive brain stimulation. So as you can imagine, focused ultrasound, transcranial, ultrasound stimulation, neuromodulation fits right within our area of interest. And it has been since we started almost 13 years ago. And even before that, when myself and Andrew, both co-founders of the company, worked together at MagStim, and kind of developed a great relationship from there. What we both have in common is our enjoyment of working with researchers and the challenges that you throw our way and trying to solve those together with then our industry partners, manufacturers. So we don't actually manufacture anything ourselves, but we hopefully are that linchpin between academia and clinicians and the engineers in industry who don't always talk the same language, literally and figuratively, to try and get the most out of what we all want to achieve. Back when we started the company, we could see actually a lot of manufacturers who would have their own distributors around the world would be focused only on their main individual products. But we could see a trend in the research field of wanting to kind of get different bits of kit working together with each other, and often that challenge was left to you guys to do, and the PhD students who wasted maybe lots of months and heartache and hours in trying to get things to work between two different bits of kit, where the manufacturers would say, no, it must be their fault. So we tried to kind of address that by being able to bring things together, things that we provide, but also things that you may have from other companies as well. That's been reasonably successful, I'm pleased to say, over those few years. So we have a great network of labs that we're very pleased to call customers and collaborators all around the world. And again, despite not being able to travel for a big chunk of time recently, we've kept those connections going and hopefully continue those fruitful relationships. In 2016, we set up the BrainBox Initiative as a way to try and pull under one umbrella some of the extra activities that we had started doing at BrainBox to support early career researchers, and that's grown now to mid-career research as well, and seeing how people develop through their academic career. In a nutshell, the BrainBox Initiative is a series of activities and events, workshops, webinars, an annual conference which just finished in London a few days ago. I think it was a few days ago. As well as prizes and awards. The Research Challenge Prize in particular useful for PhD students, postdocs who are not able to get grant funding maybe for their first big idea by themselves, but could benefit from getting some pilot data. So with that Research Challenge Prize, we're actually very fortunate to have an excellent scientific committee that do all those hard decisions for us. You know, we sponsor and make the BrainBox Initiative work, but it's all decided by you guys, your peers, your colleagues. So winners of the Research Challenge Prize get equipment on loan from us for free and support to run a pilot study, to be able to collect data, to put into a grant application or to supplement their PhD thesis. And it's since 2016, we've seen, I think now about 15 early career researchers through that process and different stages, and to see the success they get from it is very rewarding to us as well. Most recently, we've added a mid-career research prize named with the kind permission of Professor John Rothwell to again see those career paths develop all the way through. Just specifically on ultrasound, the workshops we've had to run recently have mainly been online, but we're very much looking forward to getting back to practical hands-on workshops as well. And during that pandemic, we were able to do, again, with the help of the community, put on a lot of webinars, which have been really useful as we've had feedback from the community to learn the basics of an introduction to techniques like this. So coming on to the solutions that we focus on for ultrasound. Again, some of you may be familiar of this system, Neuroscience, which is a turnkey solution that's engineered and manufactured by our colleagues at Sonic Concepts, and more on them in a few minutes. It launched only about 2.5 years ago, and in that time, we've been able to again develop it into a robust and very flexible system, and is continuing to evolve as we go, as we progress, and labs are starting to use the system more frequently now. Its probably main selling point, if you like, its main unique feature is the ability to steer the focus range electronically. It's multi-element, multi-channel, so not only can we have a single multi-element transducer and change the depth of stimulation, but also multi-transducer to focus the beam onto a more precise point. Then on the modeling side, to complement that, again, we're very fortunate to work with the developers at UCL and University of Brno with K-Plan, a graphical user interface version of K-Wave. We have it as well as the Neuroscience, we have both systems in the exhibition room next door, so please feel free to come and take a look at that if you get a chance later. And we can complement that with neuronavigation, robotics, and then the wider range of solutions that we have at BrainBox, TMS, TES, imaging techniques as well, devices that can work inside MRI and MEG. And as I said, integrating those things together is what we particularly enjoy to do, and tackle the challenge with the community to be able to deliver research that serves a purpose and not just spend most of your time trying to chase technical issues. In that last couple of years, the list of labs that are now have access to or are using Neurofus already or about to has grown really encouragingly, and using your feedback, we're able to improve the system going forward as well. Thanks for your time on that little brain box element. I'll now switch over to some slides from my colleague Kyle from Sonic Concepts. And this one's a video without any audio. So I will just jump through a few different elements of it to kind of pick up highlights. Kyle will be joining us tomorrow, so if you have any kind of burning questions for Sonic Concepts in particular, I'm not here to talk on their behalf, but rather than watch a silent set of slides for a while, I will say a few words. So again, a lot of you may know Sonic Concepts already. I can see a really great video here, but obviously you can't. What would you like me to do? Okay. So yeah, as I said, I'm not here to talk on Sonic's behalf, but Kyle will be here tomorrow to kind of answer any questions you may have. You may already know about Sonic Concepts. Hopefully you do. They do a whole range of ultrasound solutions, not only in research and medical applications, but industrial applications as well. So if there's anything about ultrasound that you're not sure of on the technical engineering side, they are the go-to people for that. If you've had the pleasure of visiting the Sonic Concepts website and trying out the transducer selection guide, you'll see that the parameters that they're working with are particularly challenging, but they have tools available that can give you an idea of like what elements you need to consider to be able to achieve the reach and targets that you want to be able to get to. We've tried to simplify that with Neurofus a little bit by giving some standard options of transducers, but as we know from research, customization is always a necessary consideration, especially when we're pushing the boundaries of what you're trying to achieve. If there's, need to go back just a little bit somewhere forward. Yes, so I'll end on this slide before Lennart pushes me off the stage like I would do in the Brainbox Initiative conference to some of you in the audience. I apologize for. I know what it feels like on the other side of the fence now. As I said, turnkey solution. We're very happy and willing to discuss with you next door and for the rest of the conference. Thanks for your time.
We have time if there are specific questions from the audience to Brainbox or Sonic Concepts. Otherwise, thanks. We'll be moving on, and I would like to invite Mark. Perfect.
Okay, so thanks, Nara. So my name is Mark Cornell from IBMT. I'm leading the transducer engineering group at IBMT. So for those who don't know us, despite the fact it's called the industry session, we're not industry. So Pronova is in between industry and research. We work for industry, we work with research, and we are a non-profit research facility. How are we structured? What is our department like? So we have 42 people, engineers, scientists, technicians, which are dedicated to developing specialized solutions in ultrasound throughout all field applications, which are thinkable, basically, from biomedical ultrasound to technical applications and even underwater acoustics. We have 5 groups. One is dedicated to simulation and advanced development. This means whenever there's a question, when someone wants to know if that's possible with ultrasound, we're going to target this by the defined simulation models, or even take the hardware we have in our labs and just try it out, see if it works. If that works out, what is the next step? Someone wants to have a solution in hand which is working, and then we can start to actually build transducers, which is happening in my group, build electronics. Build transducers, this means everything basically from single element ones to phased array or even 2D matrix arrays for all kind of applications. In the biomedical context, this can be imaging, volumetric imaging, or therapy with even 3D beam steering. Same story for the electronics. This starts at really simple ones, single channel electronics for lab equipment, up to electronics allowing 3D beam steering in a clinical context. So even certification according to medical device standards is possible. And if then at the end someone decided that, okay, that's the solution I want to have, but I don't want one of them, I want like 100, there's technologies for upscaling. So there's a group which is dedicated to upscaling of manufacturing processes, because obviously having a scientist developing one transducer in 3 months is something totally different than finding a solution for having 100 or 1,000 of them. So more precisely, in let's say the context here of the FUN meeting, what are we doing in therapeutic ultrasound? So for imaging, which is, let's say, our core business for the last 30 years, we are following a translational path from in vitro to in vivo. This means on the cell level, it can be cell imaging with ultrasound microscopes. Then on the small animal level, high resolution mesoscopic imaging systems, ultrasound, acoustic, whatever. And on the clinical side, obviously ultrasound imaging systems in 2D, 3D, suitable for in vivo human use. What does it mean for therapy applications? It starts with obviously therapy on cells. So we develop kind of cell sonication systems which allow to address cell cultures in 6 to 96 well plates, just as the one you see here. If we then make the next step and try out things on small animal, we have different single element or even matrix array transducer systems which are optimized to small animal parameters or settings. This means there is, for instance, 2D matrix arrays which can fit to the bore of a small animal PET-MR and allow you to 3D beam steering for BBB opening, neuromodulation, hypothermia. So we don't care that much about the application. We have our partners working with applications, so we don't have animal models in-house, but we have the technology to adapt things to the needs of our partners. And when it's about clinical settings for therapy, there again, everything from single element to 2D matrix arrays allowing 3D beam steering. As said before, it starts from really simple ones. We cover the whole field from imaging and therapy. It starts right at the start when someone has an idea and just wants to find out if it's possible, and we can make the final step and helping someone to let's say make the whole way until the clinical validation. So there's the capabilities for providing whole systems or components as OEM, and we have quite some experience in helping you through the certification process, which is time consuming, which can be nasty, but there's quite some experience, let's say, in testing these devices according IEC 60601, look for electrical safety. We have the test equipment in-house to make the electromagnetic compatibility, to look for the acoustic safety standards. So there's all this infrastructure, both on like what is needed in terms of documentation to comply with regulatory aspects, and let's say on the technology side to actually be able to test systems, test them in-house, and check that they're compliant with what is needed for making them usable on humans. I didn't speak about software before. I forgot that. There is one software group as well in-house. So my colleague Holger Hibner, who's going to be there. No, he's not coming because he's sick, because he planned to come. He's heading the software group, and these guys are working on all kind of signal processing tools, beamforming, therapy planning and monitoring, telemetry, and making all the software which is needed to control these systems, to tell the electronics what needs to be done, actually, put the delays in the electronics and so on. So if you're curious, we're happy to show you some systems, some transducers on the other sides, but I'm happy to answer questions right now as well.
Any questions?
Well,
Perfect.
Thanks so much. And now it's my turn to introduce our third or even fourth sponsor of the industry pitch here from Localite. You can see Julia with you there. Hi, Julia.
Hi.
Thanks so much for joining. I think we're nearly there.
Okay. Thank you, Leonard. Yeah, so hi, everyone. My name is Julia. I'm working for Localite, and I'm here to briefly introduce Localite to you, to show you a little bit what we do in the field of focused ultrasound. But most of all, I'm here to invite you to grab a drink and meet us at our booth later on, or right after this little industry session, so that we get a chance to discuss with each other and share some knowledge about focused ultrasound. So for those who don't know our company, I would really briefly like to introduce us to you. Localite was founded as a spin-off of a Fraunhofer FIT, and our headquarters are in Bonn. So we do the classical German engineering. So all the software and hardware development takes place in our headquarters. We are 100% private ownership, and we have a worldwide distribution network. We are working now for roughly 25 years in cutting-edge research. So we began with surgical navigation. Then we also did MR-based neural navigation and already navigated 3D ultrasound. And we are on one part working in the research area and developing assistance systems to work in the OR. But we also have a product line that we distribute worldwide, as I said. And this is mainly our TMS navigator, which some of you might already know. And I want to, Okay, but I quickly would like to show you our portfolio because I already mentioned we are working in the area of navigation for quite a long time now. We started with rather orthopedic navigation like our knee navigator and hip navigator. And since 2005, we are focusing mainly on our TMS navigator, where we have already developed some different kind of additions and worked in different fields. For example, we have developed a TMS navigator for the work inside the MR room, and also partnered with the French company Axilom Robotics to navigate their two different models of TMS robots. And then most recently, around 2 years ago, we started to get a hand on the focused ultrasound. And we saw this as a really promising technology, and we really want to take part in this new area. And so we decided to get a hand on that. And with the TFOS navigation, we realized that we can already support customers with our current navigation system, because of course we have some basic principles that are the same. And on the other hand, there are of course some adaptations necessary. One of the most obvious ones is of course that we have transducers instead of TMS coils, and so we want to enable planning of stimulation targets for transducers, and then of course enable the navigation of the transducers itself. We have already started to work with different manufacturers of transducers. You see a Brainsonics transducer here and a Neuroscience transducer. And yes, with this, we want to help the user to position the transducer on a desired area. And this is what we do. And for today, we want to show you at our booth an automatized transducer positioning. So for this, we also, again, work together with our partners from Axiom Robotics, and we use their TMS cobot, or let's put it differently, they adapted their TMS cobot to be able to position a transducer instead of a TMS coil, and with our adapted navigation, we can we can position a transducer fully automatically. And yes, that's what you can see at our booth. So we have the TMS cobot and we have the navigation that can show the transducer in real time. And that's how you can navigate the transducer with a cobot. And if you want to see this in motion, we are happy to meet you over there.
Oh, fantastic.
And yeah, that's it. No, that's good.
Thanks, Julia. And now for our last in-person presenter, I can see work. Hi, we have work research. We're going to prepare for you.
Hi everyone, I'm Rock Como. I think most of you know us. So I'll talk a bit about road research and how for the last 20 plus years we've been trying to help people like you accomplish their research goals. Right, so we were established in 2000. We were doing some work a bit before that, but that's essentially when I graduated with my PhD. So I was able to start a company, free and clear of the university at McGill. We're over 20 years old. We have over 1,000 labs around the world using our system. We do lots of different types of help in creating equipment that helps you. Sorry, I'm getting a little stuck here to do your work. So you know us mostly, at least on the human side, with TMS, so brain site neuronavigation system. So as I mentioned, we're at over 1,000 labs. We also manufacture the chair and other types of accessories. Right, so in addition to that, we've recently introduced a transcranial magnetic stimulator, so CTMS. So we think this is what we consider to be a next generation TMS device, which allows you to change the pulse shape, which is another way to influence whether you're exciting or inhibiting a neural circuit. We've recently finished developing our own robot. So of course we support the Axiom line of robotics, but we also, with our TMS device, have created another type of robot that we think has some other benefits that could be used for other applications. The other thing we can also, of course, add a Fluss transducer to it. So the robot was on display a couple weeks ago at the conference in Geneva. Unfortunately, we just couldn't get the logistics together to get it here for this meeting. We also have a whole non-human research side. So many of you here are probably familiar with our animal support. So we have brain sight as well for navigation, but for neurosurgical use. So in addition to the navigator, we manufacture custom surgical tools and other apparatus that you need to be able to do the experiments. So the idea is that company is very holistic in our nature. So we have experience in not just software design, but software, electronics, and hardware, so that we can help you come up with these more complicated solutions. And by looking at a problem in this holistic sense, we're able to I think, come up with more unique solutions. So in this case, we have testing chairs, training chairs that are MRI compatible, for example. We also can make custom implants. So for electrophysiology, custom head holders that integrate into these testing chairs. And the idea here, in the context of the focused ultrasound, is that these apparatuses get very complicated, right? So we can put all of this experience we have to bear to then try and solve these new research problems. So in translational research, for example, we can look at the testing chair, the head fixation that we've done, transducer support, so adding focused ultrasound to this group of other things. But typically, you're not using focused ultrasound in a vacuum, you're doing it in context of a more complicated experiment. So for us, it's a matter of introducing this new device into this ecosystem, right? And then use the navigation system to ensure that you place it accurately. right? So an example of a chair. So here we have a TMS transducer being held as well as a boost transducer in a testing chair. And here's another example of it actually in the real world. So Jerome Saleh at Oxford and later in Lyon. So this is an experiment with FUS being done in the MRI scanner. So it's not just navigation, it's a testing chair for weight behaving animals, it's MRI coils to be able to acquire good data, and holders to hold the transducer and any other apparatus. So it's this complex thing that we can put together to then solve these complicated problems. So these slides are a little slow. So here is this an example of planning in BrainSight. So you can plan where you place a transducer. We're collaborating with the group at K-Plan as well as Babel Brain to be able to incorporate these modeling platforms to incorporate that into the planning stage of the navigation. And so here's an example that came out of Babel Visco, for example. And then once you've planned it, of course, then we can use a navigation system to make sure that you've accurately placed transducer so you can carry out the experiment properly. So on the human side, it's similar in some ways. So for example, as I mentioned, we've incorporated modeling support, so Babel Visco and K-Plan. So you can use the familiar BrainSight workflow, but then you can incorporate this new capability into it. Right, so there's the planning step. In addition to that, so here's like an example using Babel Brain. Right, so the masking and then the actual simulation. And then, of course, you can display it in the software. So in addition then to just the planning part, back to hardware, we have our own chair. So we've been calling it TMS chair forever, a long time. Okay, so TMS chair, we have an arm to hold it. We can now modify it for to hold a FUS transducer. And this slide will have a transition. We've also done some smaller apparatus to hold just a small transducer in someone's head. In this case, it's a BrainSonics one. And we've developed a larger holder, custom holder for a group in Calgary to hold 128 channel transducer. So the idea is that we can look at every individual situation, we can look at what the needs are, and we can come up with a series of hardware and software solutions, put it all together to allow you to do the work that you want to do. And I think that was my last slide. Thank you.
Thank you. Thank you very much. And now we'll turn to a very last page. Actually, that's from the FUS Foundation, who've been a generous and kind supporter of both ICLAS and the FUN conferences over all of these past years. And we will record a video from them. I'll see you back in a few minutes.
We created the foundation with the vision that we were going to improve the lives of millions of people around the world with a wide variety of serious medical disorders by creating a revolution in therapy using this amazing new technology, focused ultrasound.
The Focus Ultrasound Foundation plays a central role trying to move the needle with regard to innovation.
Everybody is very passionate, is very enthusiastic, and very global.
The inspiring part of Focus Ultrasound is what it will eventually do for so many people. The lives it will save, the health it will improve for millions around the world.
We could make a significant contribution in such a wide range of medicine that it has the opportunity to really be a disruptive approach that could really transform different parts of medicine in a big way fast.
Well, time is of the essence. I mean, we're talking about people who are in need, of these minimally invasive therapies to treat things like Parkinson's or Alzheimer's.
The regulatory process in the United States can drag out for decades. Foundation holds the promise in many cases of being able to cut that short, literally by decades, if not years.
So that's why I think it's very important for us or for anyone to take on this risk to make the impossible possible.
Foundation's right in the center of helping smaller companies and research organizations really break down some of those barriers and accelerate the rate of adoption of this technology in the marketplace.
If you think about the big issues and the big problems that the world faces today, to solve them, to address them, really requires three things. I think it requires a compelling vision, I think it requires innovation, and I think it's going to require partnerships. And if you think about innovation in healthcare today, I think the Focus Ultrasound Foundation is doing a tremendous job of bringing these three things together.
They have earned the right to be an honest broker
to be the one that people look to first to articulate the vision. There are very few other institutions in the globe, I think, that have earned that kind of trust to allow them to convene and lead.
Oh, actually, beautiful video. And I'm sure they heard it all the way over in Charles. But for now, I want to invite you for drinks, I believe, and I can see that that's one of my job to do. Yeah, okay, but one thing I want to say at the very last, make sure to connect, right, with and go and play with all these fantastic toys that are over there and ask them all the questions that you can. Thanks for your attention, and now it's
Since we won't be back here in this lecture hall after this, as Leonard said, we have drinks and all the exhibition in the room across the hallway. But at 7:30, then we meet for dinner at Ice Group. I think you all have the address. If you have any trouble finding it, please come find someone with a little red dot on their name tag. And at dinner, we have drinks until 10:30, after which you are still welcome to stay, of course, but please get your own drinks. Thank you.
I have a quick question. Yeah, for the presentation this year, when people are doing their PowerPoints, are they able to see the notes version? Yes, so you can see here, for example, and see the presenter views. Good, so presenter view is there. Okay, good. So I'm gonna send, I think I'm gonna update, I was talking about it, I'm probably gonna update my slides. I'm on Wednesday morning. I'm gonna keep updating depending on what people say.